Friends, welcome to Biology Made Easy. Today in this video we are going to discuss the reproductive isolation. Now what is the ultimate aim of evolution? The ultimate aim is to produce a new species, that is evolution. But simply change in the genotype doesn't create a new species, because the interbreeding between these different genotypes should be avoided. That means reproductively they must be isolated so that interbreeding cannot take place. So that generation after generation, the variations, the genotypic variations which occur in them, they will be accumulated generation after generation and ultimately to give rise to a new species. That means the reproductive isolation is a part of evolution which leads to speciation. That is important. So, what is the main aim behind it? The main aim behind this reproductive isolation is to avoid interbreeding between the different individuals of the population. To avoid interbreeding. And any mechanism which avoids interbreeding between individuals is known as reproductive isolation. Any mechanism which separates the two individuals to interbreed is known as reproductive isolation. And the reproductive isolation is mainly of two types. It may be pre-mating isolating mechanism and it may be post-mating type. So the reproductive isolation it may be pre-mating isolation, or it may be post-mating isolation. Now in pre-mating, that means before the breeding takes place, before mating takes place between two individuals, those which are the barriers, they are coming under this pre-mating isolation. Now under this there are again sub-categories, and the first one is mechanical isolation. So what is this mechanical isolation? Now sometimes the size and structure of the reproductive organs, that means the genitals, the size and structure of the genitals, or you can tell the reproductive structures, are completely different, so that there will be no matching between the male and female reproductive organs. So in that case, the copulation is not possible. So in that case, how the organism will reproduce? That means it is a barrier. So what are the common examples? The common example of this mechanical isolation, it is commonly found in case of insects. This is a very common example. And another example you will find in case of flowers. In some flowers, the structure is so complicated. The structure is complicated so that cross-pollination is prevented. That means cross-pollination cannot take place. So this is known as, that means when the reproductive structures and their size is completely different between the male and female, this forms one barrier, that is mechanical isolation. Now the second one, is psychological or ethological. Isolation. Now ethology means behavior. That means there is behavioral differences between the two opposite sexes. Usually, in case of sexual reproducing organisms, courtship is an important phenomenon. It is an important phenomenon. And during this courtship, what happens? There are different types of stimuli responses. There are different types of stimuli responses. So these stimuli responses are very important during courtship, and that will lead to the copulation between the male and female. But if there will be certain behavioral difference between the two partners of the sexual reproducing organisms, then this is not possible. That means the breeding is avoided. Now come to the third one. The third one, commonly we can take a seasonal, or we can take seasonal, also we can take it as a temporal. Seasonal or temporal. So in case of usually what happens, the animals, particularly in case of birds, some birds they breed only in some particular season, some in rainy season, some in winter, some in summer. Similarly, in case of plants, there is a photo period. Some are long day plants, they will flower and reproduce during summer season. The short day plants, they will flower during the winter season. So how can we make a cross between a short day plant and long day plant in a nature? Artificial we can do all those things, but in nature it is not possible. So due to these seasonal differences, also the interbreeding is avoided. That is also one of the important points. Then another important point is is we can take that of the gametic isolation. Gametic isolation. Now, usually in sexual reproducing organisms, the male and female gametes, they recognize each other because fertilization is species specific. Fertilization is species specific. This is an important point. So fertilization is, when it is fertilization species specific, so each one has to recognize each other. So when they will recognize each other, if the gametes do not belong to the same species, then natural fertilization cannot take place. And another important thing also, there are some aquatic plants and animals. So they release their gametes to the water because in them the external fertilization takes place. External fertilization takes place. But when the gametes they just swim in the water, so it is not possible that anyone can combine with anyone. So in that case, since the gametes cannot easily fuse or they do not recognize each other, that is also one of the methods of predating isolation. And the fifth one under this category is ecological isolation. Ecological isolation. In ecological isolation, the organisms may be in the same geographical area, but they live in different habitats. Suppose there is a big geographical area, but some are located in one habitat, while others are located in another habitat. So that is ecological isolation. So in that case, breeding is not possible. So these are the 5 types of pre-mating isolation. Now let us come to the second point, that is post-mating isolation. So under the post-mating isolation, there are again different types, and the first one that is we can call it is incompatibility. Incompatibility. No, incompatibility means the male and female gametes do not fuse because their genes are different. That is known as incompatibility. Suppose you take the case of in some plants. In plants, for incompatibility or for sterility, there are some genes like S1, S2, S3. These are different alleles of the for sterility or incompatibility. Suppose this is the stigma and this is the pollen grain. This is the pollen grain. Suppose in the pollen grain it is S1 and the stigma is also S1. That is one of the alleles of that incompatible sterility. Then the pollen grain will not germinate on the stigma. But if they are different, suppose it is S1, but in the here it is S2, then they are compatible. Then fertilization will take place. So this is one of the important points about post-mating. Now the second point. The second point is hybrid inviability. So in hybrid inviability, what happens? Now the mating will take place. Fertilization also will take place, but the F1 hybrid cannot be formed. That means the mating occurs, fertilization also occurs, but F1 hybrid is not viable. F1 hybrid is not viable. That means the mating takes place, the crossing takes place, the fertilization occurs, but the F1 is not viable. It will not survive. That is known as hybrid inviability. Now come to the third point. The third point is hybrid sterility. Hybrid sterility. Sometimes when the hybrids are made between two distant species, the F1 hybrid becomes sterile. So when the F1 is sterile, from that again we cannot multiply that particular species. So if we'll take some examples of this case, and one of the best example will take the horse and monkey. Sorry, horse and a donkey. Horse with a donkey. Now horse and a donkey are two different species. So when a cross is made between the female horse, we can call it as mare. So when it is crossed with the male donkey, then the F1 hybrid is known as mule, but it is sterile. It is sterile. That means it cannot multiply again, because this is known as hybrid sterility. In the same case, it will make the reverse curse, the crossing. That means a horse instead of female will be male and the donkey will be female. So in that case, we'll take this as a male horse and female donkey. So in this case, the hybrid is known as hinny, and this is also sterile. So when the hybrids are sterile, how can we multiply them again and again? That means here also the crossing is not possible and new species cannot be created because the hybrids are sterile and we cannot multiply them further. Now come to the next point. The next point is hybrid breakdown. So in case of a hybrid breakdown, one step ahead we can go. The fourth point is hybrid breakdown. Now in this case, the mating takes place, then fertilization occurs. The F1 hybrid is vigorous. The F1 hybrid is vigorous. The F2 is formed. From the F1 hybrid, F2 is formed, but this F2 is sterile. Formed, but it is sterile. That means the hybrid breakdown moves to another step ahead to this hybrid sterility. Here the F1 hybrid itself is sterile, but here the F1 hybrid is quite vigorous. The F2 is formed, but it is sterile. That means the hybrids are formed, but it breaks down. So what are the common examples? The common one of the example we can take from that of the bullfrog and leopard frog. Bullfrog and leopard frog. So the F1 hybrid is sterile. So this is one example. Now let us give a second example. Example number 2. So in case of we can take the African lioness and we'll cross it. Cross is made with the Asian tiger, and the F1 hybrid is tigon. The F1 hybrid is tigon. Now if we will make the cross in the reverse way, here African lioness means this is female and Asian tiger this is male, so the F1 hybrid is tigon. Now if we will reverse it, that means in this case the tiger is female and the lion is male. So in this case the F1 hybrid is known as liger. Now these are the F1 hybrids, but when this tigon is formed, the tigon may be male and female. So this tigon may be male or female. So if it is female, or it may be male. Now usually the females are always homogametic because they have only XX chromosomes, while these are always heterogametic. They are always heterogametic, like suppose X and Y. That means the homogametic females, they are always fertile. The homogametic females, they are always fertile, while the male heterogametics, they are always sterile. Now, if the females are fertile and males are sterile, then how for the next generation, how breeding will take place between them? That means the hybrid breakdown is taking place because this is sterile. If this female has to give rise to new offsprings, then it has to be crossed with the original male tiger, Asian tiger. Then only it can reproduce. Otherwise, the hybrid breaks down here. The same case here. So here also it will give rise to female and male liger, and the females, these are always homogametic and they are fertile, while the males are always sterile because they are heterogametic. This is known as Halden's rule. This is known as Halden's rule. That means the F1 hybrid females or homogametic ones are fertile and heterogametic males are sterile, so that further generation interbreeding cannot take place between them, and they have to be crossed only with the original male one to continue this. So these are the different types of reproductive isolation, pre-mating isolation and post-mating isolation. I hope now you have a very clear idea regarding this reproductive isolation, which is very important for examination point of view. So wish you a very bright career. Thank you very much for watching the video. God bless you.